Welcome to the Deep Microbiology channel. Today, we present the final episode in our urinary microscopy series, Parasites in Urine. Let's dive in. In this episode, we will explore both common and rare parasites that can be found in urine, including 1. Trichomonas vaginalis, 2. Schistosoma hematobium, 3. Microfilaria, 4. Balantidium coli, 5. Strongyloides stercoralis, 6. Enterobius vermicularis pinworm, and 7. Paramecium. 1. Trichomonas vaginalis is a flagellated protozoan possessing 5 flagella, 4 of which are located at its anterior portion. The 5th flagellum is incorporated within the undulating membrane of the parasite. The flagella and the undulating membrane give this parasite a characteristic quivering motility. Trichomonas vaginalis is the etiologic agent of trichomoniasis, a sexually transmitted disease, STD. Many cases of Trichomonas vaginalis infection are asymptomatic, meaning that the infected person does not show noticeable signs. This is especially common in men, who often act as silent carriers and unknowingly transmit the infection to their sexual partners. When symptoms do appear, they typically develop 28 days following infection. Trichomonads most frequently represent an infection of the vagina, vaginitis, and urethra, urethritis. In females, the presence of Trichomonas vaginalis in urine is primarily due to contamination from the vagina, which is the primary site of infection. In males, however, the parasite actively infects the urethra and prostate and can be shed into the urine, especially in first void urine. Lab diagnosis: centrifuge the urine sample to concentrate the sediments, then perform a wet mount, a simple and rapid microscopic examination used to detect Trichomonas vaginalis. However, urine is not the preferred sample for wet mount diagnosis, as Trichomonas vaginalis is best detected in vaginal discharge, in women, or urethral and prostatic secretions in men. Examine the wet mount under a light microscope at 400 times magnification, and look for motile jerky moving, pear-shaped Trichomonas vaginalis trophozoites, as shown in this video. Under unfavorable growth conditions, such as changes in pH, Trichomonas vaginalis thrives in a slightly acidic environment, or high oxygen levels. Trichomonas vaginalis is a microaerophilic organism. The parasite can round up and internalize its flagella. Some believe these forms to be pseudocysts, as shown in this picture, but they are more likely degenerate forms of Trichomonas vaginalis, since they have not been reported to revert to normal motile forms. At this stage, Trichomonas vaginalis may resemble white blood cells, or renal tubular epithelial cells, making differentiation from other cells in the sediment nearly impossible. For optimal observation of motility, wet mount preparations should be examined promptly, within 10 to 20 minutes, at body temperature, as this is closer to the organism's optimal conditions in the human host. Alternatively, you can use culture media such as Diamond's medium or in-pouch Trichomonas vaginalis medium. These media are ideal for preserving and cultivating Trichomonas vaginalis, and are primarily designed for swabs and other specimens, including vaginal, cervical, or urethral samples. However, they can also be used for urine samples in specific diagnostic cases. As seen with in-pouch medium, no plating or wet mount slide preparation is needed. Simply centrifuge the urine sample, inoculate the deposit into the media, seal it, and place the pouch directly on the microscope stage for low power, 100 times observation. 2. Schistosoma hematobium. Schistosoma hematobium is a parasitic blood fluke that causes urogenital schistosomiasis. Inadequate diagnosis is potentially a serious problem, particularly in cases of chronic infection with Schistosoma hematobium that has been associated with damage to the urinary tract and eventual development of squamous cell carcinoma of the bladder. A variety of diagnostic procedures to detect schistosome infection have been reported include 1. Tests for circulating antigens, 2. Specific antibody. 3. Egg detection. 4. Hematuria and ultrasound scans of the urinary tract. Schistosoma hematobium eggs characteristics. Oval-shaped eggs with a terminal spine. Found in urine sediment after centrifugation. Best detected in midday urine samples. Collection of urine sample. 1. Exercise before specimen collection is considered necessary, so before collecting urine, patient must go up and down stairs many times, or do squats bending your knees. Why? Schistosoma hematobium eggs are embedded in the bladder wall. Physical activities such as climbing stairs or performing squats cause the bladder muscles to contract and relax, helping to loosen and release more eggs into the urine. Additionally, exercise increases circulation, which can elevate pressure in the bladder, facilitating the passage of more eggs into the urine for easier detection. 2. Collected between 10 a.m. and 2 p.m., period of maximum activity, or the reasons I mentioned earlier. 3. Collect the terminal urine specimen. The last 10 to 20 milliliters passed is required for the diagnosis of schistosoma hematobium infection, and avoid the first jet of urine. 4. If the urine cannot be transported to the laboratory for examination within an hour of collection, it is advisable to add 1 milliliter of undiluted formalin to preserve any eggs that may be present. 5. Refrigeration is generally not recommended, same as boric acid. Microscopy. Centrifuge the urine sample to concentrate the sediments, then prepare a wet mount. Start with 10x magnification to locate the eggs, then use 40x to examine them closely. Schistosoma hematobium eggs are heavy and often settle at the edge of the cover glass, so focus on the edges to find them more easily. 3. Microfilaria is very rare to be detected in routine urine. Almost all filariasis patients have microscopic hematuria and or proteinuria. Shedding of microfilaria in urine is possibly determined by local factors like inflammation, trauma, or stasis, which mainly affect the lymphatics and small vessels, causing either lymphatic blockage or damage to the vessel wall. Only few cases of Wuchereria bancrofti have been reported in voided urine sample in literature. In most of these reported cases, there was a history of chyluria. Chyluria, the presence of chyle in the urine, is caused by rupture of dilated abdominal lymphatics into the urinary excretory system. Chylous urine is milky in appearance, and blood is sometimes present due to rupture of small blood vessels into the urinary tract during the formation of lymphatic-urinary fistulae. As you can see in this picture, the urine is milky white, and here it is milky white with a pinkish color, which indicates the presence of red blood cells. For the detection of microfilaria in urine, pour 15 milliliters of urine into a conical centrifuge tube and centrifuge for 5 minutes at 350 G or higher. Discard the supernatant and examine the sediment microscopically for the presence of microfilaria, as shown here in this video. 4. Balantidium coli is the ciliated protozoan which is known to infect human and non-human primates. Route of infection is fecal-oral route. It is actively motile and causes mostly asymptomatic infections, and it may develop dysentery, which is similar to that which is caused by Entamoeba histolytica. Extraintestinal balantidiasis is rare, but has been reported in several organs, such as the liver, lungs, and genitourinary tract, in immunodeficient and otherwise healthy patients. Urinary balantidiasis has been reported rarely and mostly in immunocompromised patients like diabetics and renal transplant recipients. Here, you can see that the Balantidium coli trophozoite is covered with cilia, which allow it to move actively in liquid environments like urine. The macronucleus controls most of the cell's functions, while the micronucleus plays a role in reproduction. The cytostome, essentially the mouth, is where the trophozoite takes in food. Balantidium coli trophozoites can be detected in urine sediment after centrifugation. As seen in this video, The trophozoites exhibit high motility. 5. Strongyloides stercoralis. An intestinal helminth that infects humans through contact with soil containing the larvae. Disseminated strongyloidiasis occurs when larvae invade organs not classically associated with the normal life cycle of the parasite, including urinary tract. And it occurs in individuals with a weakened immune system, such as those with human T-cell leukemia virus type 1 infection, prolonged corticosteroid use, organ transplantation, HIV/AIDS, or malignancies. Detecting Strongyloides stercoralis larvae in stool, sputum and gastric juice is diagnostic, but rarely reported in urine. And here, you can see the motile larvae of Strongyloides stercoralis in the urine sediment, actively moving under the microscope. 6. Enterobius vermicularis pinworm is an intestinal nematode of humans. Mode of transmission is fecal-oral. Human infestations occur when the eggs in the infective stage are accidentally ingested. The majority of infestations are asymptomatic, but ectopic infections in the genital or urinary tracts do occur. Here are the ova of Enterobius vermicularis found in urine sediment. The presence of eggs could be due to contamination from feces or an ectopic infection. 7. Paramecium is a unicellular ciliate protozoa. Paramecium species is visible to the naked eye because of their size, 50 to 300 micrometers long. Microscopically, it is slipper, cigar, or spindle-shaped. The entire body surface is covered by cilia. Endoplasm is semi-fluid and less granular. Is non-pathogenic to humans. It is found in fresh water including ponds, rivers, lakes, streams, and pools. Can come from contaminated water. A few documented cases mention that paramecium has been isolated from urine, and in some of these cases, it has been associated with pathogenic infections. And here, you can observe paramecium in the urine sediment, and you can notice its size compared to other cells in the microscopy field. Thanks for watching. If you enjoyed the content, don't forget to subscribe to Deep Microbiology channel and turn on notifications to stay updated.